organized by the Ruhuna Medical Laboratory Science Student Society. Today, we are diving into a topic that many of you have been eager to explore: unlocking career opportunities in the UK.
Absolutely. This session promises to be a gateway to new horizons, shedding light on the paths available for MLS graduates aiming to build their careers overseas. To guide us through this journey, we are thrilled to have a distinguished speaker who will share her experiences and wisdom with us.
To formally open today's session, I would like to invite Mr. Ashant Satsarar, the President of the Ruhuna Medical Laboratory Science Student Society, to deliver the welcome speech. Mr. Ashant, over to you.
Good afternoon, everyone. It is both an honor and a privilege to welcome you all to the second session of the webinar series organized by the Ruhuna Medical Laboratory Science Student Society. I would like to begin by extending a warm and special welcome to Professor Imendra Kottapola, Dean of the Faculty of Allied Health Sciences, University of Ruhuna. Your presence here today is truly inspiring and greatly appreciated, sir. Although Dr. J.A.N. Sandamali, the patron of our society and the head of the Department of Medical Laboratory Science, is unable to join us today, I would like to take this moment to acknowledge her unwavering support and encouragement for our initiatives. A very special welcome goes to our esteemed guest speaker for today's session, Ms. Dananjani Rathnayake, genetic technologist at Cardiff and Vale University Health Board, who has kindly accepted our invitation to share her knowledge and expertise. Additionally, I wish to acknowledge and welcome our valued academic staff members, including Dr. Sachinthi Amarasiri, Senior Editor, and Dr. Iranga Silva, Senior Treasurer of our society. I also warmly welcome all my fellow undergraduates from the Department of Medical Laboratory Science who are joining us today. On behalf of the society, I warmly welcome you all to this session. This session promises to be an enriching experience and encourage all of you to take full advantage of the opportunity to learn and engage with today's insightful presentation. Thank you for your time, and let's make this session a grateful success together. Thank you very much.
Thank you, Mr. Ashan, for that warm welcome. We are now honored to have with us Dr. Sachinthi Amarasri, Senior Lecturer and the Senior Editor of the Ruhuna Medical Laboratory Science Student Society, Faculty of Allied Health Sciences, University of Ruhuna, who will share her thoughts on today's session. Madam, the virtual stage is yours.
Thank you. Good afternoon, dear sir, madam, and my dear students. So this is the second webinar organized by the Ruhuna Medical Laboratory Science Student Society for the year 2024. So as you all know, one of the main objectives of the Ruhuna Medical Laboratory Science Student Society is to give students an idea about how their professional careers can be developed beyond the role of a conventional medical laboratory scientist. So I hope you will use today's webinar as a good opportunity to identify and understand a specific area of interest. So I would like to make this an opportunity to thank Ms. Dananjani Rathnayake, a genetic technologist at Cardiff and Vale University Health Board, London, for accepting our invitation and being the resource person for today's webinar. So I wish all of you all the very best for today's webinar, and hope this session would be a very fruitful session for all of you. Thank you.
Thank you, Madam, for your encouraging words and continued support. Now it's time to dive into the heart of today's session. It is our privilege to welcome our guest speaker, Ms. Dananjani Rathnayake, a genetic technologist at Cardiff and Vale University Health Board.
Ms. Ratnayake's journey is a remarkable one. Armed with an MSc in Medical Genetics from Middlesex University, London, and a BSc in Medical Laboratory Science from the University of Sri Jayewardhanapura, she has forged an impressive career in the UK. Her achievements and expertise offer an inspiring example of the doors that can open for MLS graduates abroad.
Before we begin, I would like to remind you all that at the end of this presentation, we have arranged a Q&A session. So you can unmute your mics and ask questions regarding today's topic from any language that you prefer. And also, we will keep the chat box open from our resource person. Until then, please make sure to keep your mics muted. Now, let's give a warm virtual welcome to Ms. Dananjani Rathnayake. Over to you, Ms. Rathnayake.
Thank you very much, all. Thank you very much for giving me this opportunity to talk to you. And also, thank you very much for the warm welcome. I'm going to try and share my slides now. Okay, before we continue, I would like to ask from the audience we have today, I want to get an understanding about who we have today. Are you all undergraduate student, or like is there anyone who is already working, or are you all first years, second years? Can anyone put in chat or unmute and introduce yourself or tell? Okay, I will assume most of you would be still in undergraduate process.
Miss Dananjani, majority is undergraduate, and there are some academic staff as well.
Amazing, amazing. Thank you. Okay, in my first slide, I'm going to talk about how different the healthcare system here in UK compared to how it is in Sri Lanka. So my first thing is I'm going to talk about the structure. Structure-wise, it's mostly the same. So when I work in Sri Lanka, we work in hematology, biochemistry, microbiology, and different departments. So the structure-wise, the healthcare system here in UK is the same. So we have biochemistry, hematology, genetics, and everything. So you might see some differences. Like when I was in Sri Lanka, when I work in my lab, we had a flow cytometry department under hematology, but here we have a different department for flow cytometry, and hematology is a totally different department. But I think you can understand it's same structure. The second thing is training and qualification wise. So this topic, I'm not going to go into in detail in this slide because I'm going to explain it later properly. So, but basically, we need a degree in biomedical science or related field. Also, we need the registration. It's from Health and Care Professional Council. It's CPC registration, and that is a mandatory requirement. The next thing is research and development. Most of the clinical lab in UK, they collaborate with researchers and mostly clinicians with their research and development programs. So example, in my lab, we collaborate with, so I work in a genetic lab. My lab, we collaborate with clinicians who treat cancer patients, and those clinicians, they have access to most recent drugs. Some drugs are not available in the market, but from the research, they have the access to the drug that can treat the cancer patient. So they choose a cohort of cancer patient and they trial those drugs. So in that process, we provide different test method, and we give different information to those clinicians and researchers. So that is really interesting part of our day-to-day life. The next one, the career pathway and specialization. So in my experience, in Sri Lanka, we don't have a tier system. Of course, we have senior staff and junior staff, but that depends on how many years you have work in that particular area or how many years of experience you have. But here in UK, we have a tier system. So it means, so it starts at band 2. So we call it a band system. The lowest band is band 2. The highest band is band 9. So 2 to 9, we have a certain type of qualifications you need to acquire and certain level of experience you need to have. So you don't, sometimes if you have right qualification, you don't have to start at most junior level. You can start at mid level and progress within your career. So those details I'm gonna explain in next few slides later. So my focus today is mostly about the diagnostic service. So mostly, as you may already know, diagnostic service, laboratory professions, we work in private or government hospitals and some clinics. Then I'm going to talk about the nature of the duties we have. So in most of the lab, we have a technical team and we have a clinical team. So duties of technical team involve like they receive the sample and they process the sample until the results is out. The clinical team, after results is out, they do the analysis for this testing, and then they see if this results, does that make any sense clinically? So that is the duty of clinical team's responsibility. So in technical team, the technical team involve a lot of medical laboratory assistants, medical laboratory technicians, and biomedical scientists, and also some clinical scientists as well. In clinical team, we have some technicians, and mostly it involved with clinical scientists. The next thing, the grading system. As I mentioned earlier, the grading start at band 2. So this is the entry level. As you can see here I have mentioned the salary range. This salary can be a little bit different now because the government recently changed the salary threshold, and also salary can be a little bit different based on where you work. Especially if you work in higher cost areas such as London, you get higher salary because it's a high cost living area. So this is the national salary range for now for the band 2 medical laboratory assistant, and this is entry level. Qualification wise, there is no formal qualification needed, but basic healthcare experience might be useful, but this is the entry level. So mostly the band 2 technicians or assistants, they work in sample reception, and few of their duties involve accepting the sample, and then they can see if the sample is in right condition. Is this sample right for the the indication on the request form. And then after everything is matched, after the sample is triaged by a clinical scientist or biomedical scientist, they enter this sample to laboratory information management system. And then they also help distributing this sample from sample reception to the relevant labs. And also their duties might involve some data entry and also sample or region stock management as well. So the next one is band 3, that is medical laboratory assistant. This is a bit higher level than band 2. For qualification wise, some relevant experience or like a diploma in healthcare science might be asked when they advertise the band 3 jobs. They might do a little bit of processing of the sample, like they will prepare the sample based on different requirement of the testing process. The qualification wise, even though the formal requirement we need is diploma in healthcare science or diploma level qualification or experience, most of the time, in practically, now I feel like most of the people have gained degrees and they start at even band 3 levels. And I think that has made the job market a little bit competitive. The next one is band 4. They would call technical officer or band 4 medical laboratory assistant or associate practitioner. They might, qualification wise, they might need a foundation degree or equivalent experience. So duty wise, they might perform the testing under a qualified biomedical scientist. They have more responsibilities than a band 3, obviously. So the band 5, so this is where you need a degree in biomedical science or related field. And also you need to have the registration in HCPC as a biomedical scientist. Some hospitals, they provide entry in because this is the entry level. Some hospitals allow you to join their team as a band 5 if your registration is ongoing, but normally the requirement is you should have the registration as a biomedical scientist to get a band 5 job. The next one is band 6. So this is a specialized biomedical scientist or pre-registration clinical scientist. The qualification wise, in this stage, we need the postgraduate qualification, or also like first you start as a band 5, then you get a specialized training, and then you can progress into a band 6. So when you come to band 6, you are more specialized. So let's say you start in biochemistry, but in biochemistry lab, you choose a different pathway or different test type or different disease type to progress with, and then you become specialized in those services. So next, band 7 and 8. At band 7, they already have the registration as a clinical scientist or biomedical scientist, and they are like the most senior people in the lab you always would be engaged with in the lab, and they oversee all the diagnostic tests and they manage the team. And in band 8, so band 8 is divided into subcategories from band 8A to band 8D. So this level, you have to gain special qualifications or fellowships, which I'm really going to talk later, because this is not going to be an entry level requirement you would need. But they would be laboratory managers and they always have postgraduate qualifications and fellowships. So at band 9 level, so that is the consultant level. So the job title is consultant biomedical scientist, and then that person become the head of the department. So this FIBMS fellowship or FRC path, these are the two qualifications you should gain to achieve a band 9. So in the lab, you would see only one band 9 because that person would be the head of the department. But like in band 7, you would see 5 or 6, a lot of band 7s. So this is how everything is divided band wise. I have a few more details which I'm not going to read about here, but I would say share the slide. You can have a look if you are interested. So now I work as a band 5 genetic technologist. In next few slides, I'm going to talk about few of the duties I do, because then you can understand the level of responsibility you can have as a band 5 technologist. So one of the analysis I do is karyotyping. I think you all might be familiar with what karyotyping is. Would anyone mind telling me, is this a female patient or male patient? The disease type is CML, chronic myeloid leukemia. You can put in the chat. Great. Male, yes. So that means you have understanding about what karyotyping is and everything. Would anyone mind guessing if you see any abnormal chromosome? So you would compare. So we have one chromosome from our mom, the other one from dad. So we compare how they look, each band. So we have pale area, dark area. So you're going to compare how they look next to each other. And do you see any abnormality? This part is just a bit of a stain. So please ignore that part. So our expected abnormal, oh yeah, yes, great. 22 is abnormal. Part of the 22 is gone. There's another chromosome which is abnormal here. Can anyone guess? So this missing part is somewhere in another, yay, 9. Great. So here the chromosome 22 is deleted. So that deleted part is just here. So this is a typical abnormality we find in CML patient. Thank you all. This is how we write the chromosome abnormality. So this one, I'm not going to explain a lot. So this is a APML patient, acute promyelocytic leukemia. What happened here is part of chromosome 17 is deleted and it's combined, translocate to this chromosome 15. And the edge of the chromosome 15 is here, which actually you really can't visibly see. And we use another test type to confirm this translocation. So I have put some information about APML and why this testing is really important. But you can have a read, but I'm not going to explain because it's not the purpose of our seminar today. So one of the other tests I do is fish analysis. So what you see here is a metapase, so all of your chromosomes. So in this fish analysis, we are going to look for a translocation of chromosomes like we saw here from chromosome 17 to 15, 15 to 17. So they are sharing genetic material. So that's what we are going to test here. So in a normal cell, I hope you have understanding about fish. Fish stand for fluorescent in situ hybridization. So what we do here is we have this red fluorescent label probes. These are complementary to the chromosome 15, the breakpoint area, and the green fluorescent label is for chromosome 17. So if this is deleted or translocated, we can identify the pattern. So in a normal cell, you would see this pattern because there's no translocation. It's 2 red signal and 2 green signal. So this is a nucleus. This is a metapase. So in this nucleus, everything is inside the inside, and you see here you see a translocation, especially in this one. It's clear. So if it's a translocation, because they share the genetic material, translocation happen. You have both green and red together. So when green and red is together, it resembles like a yellow. So this is an abnormal nucleus. So here I have listed another test. To be honest, I'm not going to explain this because this is going to be a long process explaining all the principle and stuff. But this is a pyrosequencing method. So, and you can have a read through. And this one, I'm going to talk a little bit about sequencing. So for different cancer type, we get their cancer samples. Sometimes they are blood sample. Sometimes we get a FFP sample from histopathology labs. So what we do is we sequence, and then we are trying to see if they have an abnormality that we can treat it for. So especially in melanoma. So we have guidelines. We call them NICE guidelines. In melanoma, we are looking for mutation in BRAF gene at codon 600 level. So if we see a mutation in BRAF gene at codon 600, we have treatment available. So to treat the patient based on their mutation they have, we do this NGS testing, the sequencing, and this is how we display the sequencing, the mutation in our software. And this is how a report would look like. And we give recommendation about, like, we explain this is the abnormality, a mutation we saw, and based on this abnormality, we do a literature search, and then we see what are the current treatment available, and then we give them treatment. We give the clinicians the treatment strategies they can use. Okay. Next, I'm going to talk a little bit about emerging trend in cancer genetics. So my purpose of explaining this, the emerging trend in genetics, is not not just to understand trend in genetics, if you are applying for a job in any field, it is really important you to do like a good read, good research. Let's say you are applying for a biochemistry lab. You need to, before you go to interview or before you apply for the job, it's really good to have like a good understanding about emerging trend in your interested area, because in the interview or even in your application you can show them you are passionate, you are up to date, and that's that will make you really highlighted within the team. um, so I'm not gonna talk about everything, but I'm going to talk about two different things. The first emerging trend I'm going to talk about is liquid biopsy. So normally when you have a surgery, what we all know is we go through a surgery, and then after the surgery, this sample or the surgical parts go to lab, and then in histopathology labs, histopathologist decide is this patient have a cancer, or is it what type of cancer, in what grade, so everything. But in liquid biopsy method, the principle of the testing, the basic principle is when you have a cancer cell in your body, when that cell die, all the genetic material get mixed with blood and it circulate in your body. So we take the blood from that patient who has the cancer, and then we separate the blood, and then we see if this patient has this particular mutation we are looking for, this cancer. So you would get a diagnosis or prognostic indications even before going to the surgery. So this has been really helpful. But the thing is, this test is still at really early stage, and a lot of literature and a lot of research needed to support this testing. But it has become really interesting and emerging trend in cancer genetic and diagnostic service. And the next one I'm going to talk about is immunogenetics in cancer patient, mostly the treatment process. So the basic principle is, so you have your healthy cells, and the healthy cells has certain genetic material, and those genetic material decide um, what kind of protein you express in your cell membrane. So same way, your cancer cell has slightly altered genetic material. Of course, that's why it's a cancer cell. So because of that reason, the protein this cancer cell express is different than to the normal cell in your body. So if I have a cancer, they take part of my cancer cells, and then they do all the testing to identify what are the proteins that I have in my cancer cells compared to my normal cells. And then they develop vaccine just to target these cancer cells. So this vaccine only gonna target antigens on your cancer cells. So because of that reason, this is highly personalized and highly effective method. And because of this cancer vaccine, patients get really better quality of life because they don't get much side effect as other chemotherapy drugs, because chemotherapy target all the cells, not only the cancer cells. But in this way, this developed immuno-based vaccine for these cancer patients, they only target cancer cells. So this is mind-blowing, really emerging trend in cancer genetics now. So all other things, I'm not gonna go into details, but of course you can have a look. So next slide. I'm gonna talk about where you can find jobs. The first one I'm going to talk about is STP, So this is a scientist training program. That is what STP stand for. So I'm going to go into this link. So in this page, what we are going to be interested is scientist training program, applicant. So before I continue, I'm going to give a little brief information about this. So this is a scientist training program. If you are not a scientist, they train you to be a scientist, and anyone can apply for this one. Because of that reason, this has become really competitive thing to get, but it's still not totally impossible. So the structure of this program is it's a 3-year program. They recruit you, the NHS, the National Healthcare System, they recruit you as a band 6 trainee scientist, and then they train you for 3 years, and they pay you the salary for band 6 scientist. And finally, after completing these 3 years, within these 3 years, so this is hospital-based, the work-based program. And what you can do is they collaborate with a university that can provide the coursework for you. And then at the end of this 3-year program, you get a master degree, and also you can get your registration as a clinical or scientist. So that the main advantage in this is you get paid for 3 years. It's like you get the work-based experience, and you get a master degree for free, and you get the registration to work as a scientist. So this is the scope of this program. So here, every year around January to March, they publish all the information and date in this page. And then what you have to do is they put all the link related to this, and then you can create an account within this website, and then you can apply for that. And then if you are selected, the selection process is few steps. So first step, they will select your application, then you would have to go for an interview. So mostly it's online interviews. If you decide to try this application process, this route, it is really important to put your good effort into this, because this is really competitive stream to get in. Okay, back to the slide. So next one, so track job is the next one. So this is where they mostly advertise the healthcare jobs. You just have to create an account and then log in. So I think I have already logged in. So after you log in, you can search for any jobs. So let's say I'm gonna search for biomedical scientist job. So then you would see all the jobs they have been advertised, and you can filter them. So based on band, then you can see your interested jobs. So okay, I'm gonna open this one. So when you open one, you can see all the information, like they give you all the information you need to know about the job. So it's a band 5 job, the contract is permanent, and this is the hours you have to work. You might need to work in shift pattern, and where you are going to work, like all the details you would need. So when I tell you this is the salary you get, this is not the monthly salary, this is the annual salary. So in the application, the advertisement, they give you overview about how the job is and what you would be doing, and then they will list your main duties, and they will give you some information about their values and what we would get from working to their organization. So here they put some essential criterias and desirable criterias. So essential criterias are the criterias you should have before you apply for this job, because this is a biomedical scientist job. They need you to have the registration. Then they need to have the accredited good degree in biomedical science. So desirable criteria, you can still apply, but based on the application load they get, they will prioritize and they will see, but they would prioritize your application if you have fulfilled their desirable criteria. It's the same for experience and skills. So they would expect you to have this essential experience, but desirable, probably you can have them, or if you don't have them, that it doesn't matter, you can still apply. So what I want to highlight in this part is, normally there's a few documents you can download. So in these documents, they tell you what are the duties you have to fulfill and what kind of qualification you should have. So after you start applying online, what you have to do is you have to fill certain you have to create an account, then fill all the information. So filtering level, what they do is they have a marking system. So let's see, so they mention so experience wise, they need these things. So based on the experience or based on qualification, they expect you to have, they have a marking system. So you really have to show off yourself. So let's say evidence of team working. So you need to use this word team working and then explain how you have proven experience or proven evidence that you have worked in a team and how you have shown a team working. So when you apply, just read through everything and then highlight everything and then try to use those words and try to build a good application. The highest qualification, you wouldn't get shortlisted or anything. So that's my advice there. So same process, NHS job, you only have to create an account and sign in. Then same process, you can start applying for jobs, and this is how the website look like. So if you are interested working in blood bank, this is a link for the blood bank jobs. So what you have to do is see our vacancies, and then you can filter jobs based on what you want to do, and then you can start applying for those blood bank jobs. Mostly here you might find these jobs sometimes in all other platforms as well, but mostly these are the main official sites. The next thing I want to talk about is STPE. So this is kind of similar to STP, and what STPE stand for is, this is Scientist Training Program Equivalent Process. So in this process, it's a band 6 job. You work as a pre-registered clinical scientist or pre-registered scientist, and you get 3 years, and then you gain work-based evidence, and then you apply for your registration. So the only difference here to the STP is you won't get a master's degree at the end of this, But the most important thing is you get the registration as a scientist. Okay. Next, I'm going to talk about hot topics in interviews. The first one is quality management and accreditation. So it won't be this order, but you might see these questions also in different bands. So from band, if we apply for a band 2 or if we apply for a band 6, the level of the depth of the question they ask can be totally different, but this is the general understanding you should have. Of course, you should have a good read on all these topics, good understanding. But this is the overall. So they might ask you about quality management and accreditation. So in UK, the accreditation body we have, we call it UCAS. So that is the accreditation body. All the accredited lab, they have the registration in UCAS. So as you all know, the laboratory standard we should have is ISO 15189, the ISO standard. So the under the quality management, it is important to have accreditation, external quality management, and internal quality management. The lab I'm working is most of the tests we enroll NECA's external laboratory EQA programs. So having an understanding about external and internal quality control is really essential. So in some jobs, especially if it's a technical job, they would ask you about technical question. So what you can do is, let's say if you apply for a genetic lab, you can go to their site, their website, and everything is on the internet. Then you can have a good read on their website and see what kind of tests they are doing, and then you could get a better understanding or polish your memory on the principles. And if you have experience in those areas, you can try to remember the troubleshooting events you did and your certain experience you have. So when I say experience, let's say they ask you like, do you have experience in karyotyping? So then you would straightly think like, oh, I haven't done any karyotyping in a clinical setting. You don't have to say that. What you have to say that you know about karyotyping. Sometimes probably you will have like a you have seen someone doing karyotyping and you have the basic understanding. So before telling that, oh, I don't have any experience, tell them like, oh, even though I haven't done any karyotyping in clinical setting, I am really interested. I have done these things and I have learned these things in my undergraduate program and in my final training, the hospital training sessions. I witnessed these things, and this is the level of experience I have, and I'm really motivated to learn these things. So you can show how interested and inspired you are. Like every negative thing, you can turn it to a positive thing. So next thing, they might ask you some details about clinical aspect of some diseases. So this question, they might can be specific for some clinical jobs. So as I mentioned earlier, we have a technical team and we have a clinical team. Of course, sometimes in some setting, there's an overlap. Technical team do clinical stuff, clinical team do technical duties. There can be overlap, but most of the time there are two groups. So in if you know in your job description, if you know you might be doing some clinical duties, it's better to have an understanding about some clinical aspect. I'm sure like I not I'm sure I know that your undergraduate program is really advanced and you really get really good knowledge around most of the stuff you should know. So in clinic, again to this one, so they might ask about like, if it's a cancer lab, they will ask like, oh, can you describe one type of cancer, probably a pathophysiology, and you don't have to give like a 2-page description, just 3-4 sentences, and then because our jobs are laboratory-based tests, they might ask, you can explain. So for this test, these are the tests for this disease, and this is why this happened, and these are the test method we use as a lab to test this testing, and this is how it's going to be important for patient. If it's too confusing, I can give you an example. So let's say I'm going to talk about melanoma. So you can tell how melanoma happened, what is melanoma is. So it's a skin cancer, this is how it happened, and things. And the most important test we do to detect melanoma is in my lab is in genetic lab is sequencing. So the test method we use is sequencing, and then we look for certain genes, and then if we see this gene is abnormal, how this is going to affect the patient is for melanoma patient, if BRAF gene is abnormal at codon 600, this patient can get the certain drugs, and that is the advantage of doing this test. So to that extent, so you might don't need everything, but having an understanding is really important. So next one, there can be a question asking how you prioritize your workload, because they really have to see if you can work and if you can take the responsibility for the work you are doing. So there can be some questions about how you prioritize your work. Always try to remember, when you prioritize your workload, you had always remember not what easy for you to do, what is most important for the patient, how my decisions gonna impact the clinical decisions that made on the patient. So how this is going to be affect or advantage or disadvantage for a patient. So one of the question, so this one, the trust values. So this is something I wasn't very familiar either, but they always ask one question. So different hospitals, they have a different value set. So what I can say about this is all the qualification. Nowadays, everyone has qualifications. Everyone know everything. AI is here. Everything is on the internet. Now, the most challenging thing in a team is to find the right fit, so right person with right qualities. So different hospitals has different value set that they want to work on their team. So I will explain it by this. So I'm going to go to this link. So this is the hospital I worked before. So their values is welcoming, and these are the list of their values. So they might ask you, could you tell 3 or 4 values you saw on our trust, our hospital? So then you can say, if you have done your homework, then you can say these are the trust values. So then they would ask you, oh, okay, you mentioned this trust value, let's say respectful, and then could you explain a situation that you show respect in your workplace? So things like that they could ask you, because it's really important you to do your homework and build up on these answers because, of course, English is not our first language, most of us. And then sometimes, you are respectful, but you might go blind if you haven't prepared yourself for an answer. So that's my advice on that. The next one, the last one I'm going to talk about is, finally, they always ask, like, do you have any questions? So most of the time, personally, what I was doing was, when they ask, like, do you have any questions? I'm like, okay, I am in this stressful interview environment and everyone is looking at me. I just want this to be finished. And then I really don't want to ask any questions. That's how I was. But that is really not the way to do it. What you should do is, when they ask you, do you have any questions? This is the place you can show that you are passionate, you are interested, you are the right fit to their job. So this is where, so as an example, let's say, so you apply for a genetic job and then then you check their website and they have mentioned in their website, oh, we are going to implement whole genome sequencing within next 2 years. And then if you saw that, what you can do is you can ask some question about their project. You can show some interest, and then they would know, like, you have done a great job, like looking things, and you are a really interested person, and then you are going to be really highlighted. So that's my advice there. Okay. The next one, I think I once mentioned something about NICE guidelines. So I'm going to go to this web page. So NICE guideline, this is a website. NICE guideline stands for National Institute for Health and Care Excellence. So this is where all the research and all the clinical and all the trials data is gathered. So for all the diseases in UK, we can see all the guidance for testing and diagnosis and patient management. So this website is not only for laboratory staff, so all the researchers, all the clinical staff, all the doctors, consultants, they refer back to this website, and then they see the latest cutting edge researchers and everything. Then these are the where all the recommendations are for patient management. So let's say my favorite, hope I never get it, melanoma. So you can see, like, they say, like, how you are going to assess and manage, and how you're going to test, what kind of drugs you can use, and all the latest information is here. So the purpose of today, I'm introducing this to you, is not to show everything on this page, because personally, I'm not familiar with everything in this page either, because it's too much overwhelming amount of information. What I want you to know is all the guidelines we have about diagnostic and patient management is come from here. So this is the site we refer to. The reason we refer to this site is this is where all the this is a big team working in this organization, all the clinicians and researchers and everyone, and these are all the new information is available. Okay, the next slide. Okay, now I'm going to talk about test directory. So this is test directory is for doctors who write the test, who request the test, and for us as a laboratory staff to see if this is available. So I'm gonna show you. When I show you, it will make more sense. So all the disciplines, we have different test directories. So this is the test directory we have. I use in my lab. So I'm going to download this. So this is the Excel sheet, very crowded. So let's say I want to, so because I download the test directory that is relevant to cancer. So you can see it's solid tumor, neurological, and hematological, and everything. So now if I say I want to know about melanoma, I can just search the melanoma and see melanoma in adult. So these are the test name doctors can request. So in each test, so if a doctor requests multi-target sequencing panel, these are the three genes relevant to this cancer we should test in our laboratory. So this should be our standard as a lab. So here we give you all the information, extra more information, which I'm really not gonna go into detail. You can have a look if you think it's interesting. Okay. So my purpose of showing you that is all the disciplines for hematology, for biochemistry, all the disciplines, they have this test directory. And if you know you are going to a lab, you are applying for a lab, that do a certain type of disease testing, you can find the test directory and you can see what kind of test you are doing. Then you can get a proper preparation. Also, this is really useful in our day-to-day duties as well. If you get a request, let's say you get a request for melanoma, but the doctor has requested a totally different test type, then you can tell them, okay, this is not what test directory tell us, this is not the indication for this test, we can't accept this. So you have a guideline back to refer to. So here I have put some quality management few things, but I'm not going to explain these things now because you can have a nice read, because I know you all are familiar with these terms. The next one, I'm going to talk about qualification we could gain to work as a scientist. So the first one is IBMS registration portfolio thing, but the IBMS is more like a assessing body. So people get so confused with IBMS and HCPC. So now I'm going to talk about biomedical science side mostly, because that's where we all are interested. So this is IBMS side, and this is the HCPC side. So different in these two. So here we have the link, you can use them. So IBMS is more like an assessing body. They assess you have the right qualification to get the registration. So the HCPC is the registration body. They register you for your right qualification. So most of the degrees in Sri Lanka we don't have to go through IBMS assessing part, because most of your degrees, all the degrees in Sri Lanka, when we get our biomedical medical laboratory science degree, it has all the component needed to get HCPC, and there is a very low risk of getting rejected. Actually, I would like to appreciate the opportunity that I got to study in Sri Lanka in our university, because I studied in Ruhuna for 3 months, and then I transferred to Japura. But during both times, I realized like the depth we are learning, and our course work is absolutely amazing. And I think you should be really proud of yourself, because so when I work here, when they say something, I feel like I have really good understanding about what they are talking, because we learn about hematology, we learn about biochemistry, microbiology, or the molecular biology and laboratory management. We learn everything. So most of the people I met here internationally, sometimes they only have done sometimes neurology, sometimes just human biology. Like our degree is really amazing one, and it touched most of the areas, and it's really solid foundation for you guys. So because of that reason, you really don't have to, it is not necessary to go through a assessing body, because your degree is going to be accepted in HCPC. But sometimes some people have like an agricultural degree, and now they like to work in a biomedical biochemistry lab, and they want to get registration as a biomedical scientist. Now they can go through this IBMS assessing body, and then you can provide your coursework and all the information they ask for, and then they will assess your degree, and they will tell you what are the sites that you haven't covered, and they will recommend you some courses you can do. So after finishing those courses, after fulfilling all the requirements they ask you to do, then you can go for the HCPC registration. So these are the documents they ask you to bring for the registration, and they have an international route and another route for the local students, but these are the things they might ask you to bring. So academic qualification wise, if you work in healthcare sector, having a bachelor degree and a master degree, it really helps. Sometimes some people I met, they have a PhD, but if you work in healthcare system, the government healthcare public healthcare system, it's not essential to have a PhD. But if you go for a private hospital, not the hospital, private organization like AstraZeneca or something like a big company, they might ask you to have a PhD in certain areas they are interested. So let's say you want to go to AstraZeneca, they might ask you a PhD in immunogenetic or immuno-based PhD, because they prepare like vaccine and stuff. So that's different in private companies and biotechnology companies, but in government hospitals, it's mostly having a bachelor. Even having a bachelor is enough, but you can then progress, and when you progress after band 6, you might need a master degree or postgraduate qualification. I think I talk about this. So yeah, so in this slide, I'm just going to talk a little bit about genetic technologies, because it sounds a little bit confusing, because we have a and it's not very familiar for people. So these are the two registration that you can get if you are willing to work in genetic field. So the first one is Academy for Healthcare Science registration. So this is for genetic technologists. And if you work in technical team, this is really good to have. And then if you work in clinical team, having this does not hurt, but you might have to progress to get the HCPC registration. And during the HCPC registration level you can achieve is a clinical scientist. Okay. So I think this is my last slide. So as I mentioned earlier, after band 7 or when you go to band 8, you need to have this fellowship or FRC path. So one of these is essential to have when you go for higher bands, but at your level, it's not even for me, this is not relevant for me. So this fellowship is given by the IBMS, but the FRC path is Royal College of Pathologists. So to achieve this one, this is more clinical side, and this is more technical side, but they all get paid same. It is just your interest. Do you want to work in clinical side? Do you enjoy working with samples, analyzing and stuff, or if you just enjoy doing and only the analysis part of the testing. So with this fellowship, this is not for this. It's a it just show you a qualification level, and it's a fellowship. But this FRC path, it's more like you need to face some exams and get training, because end of this you get the career level of consultant pathologist, and for the fellowship you get the level of senior biomedical scientist. Here I have mentioned a few things. I think that's the end of my slides.
Thank you very much, Ms. Dananjani Rathnayake, for taking time to be with us today and for sharing such valuable insights. We are truly grateful for your presence and the wisdom you have imparted. We now open the floor for questions. This is your opportunity to engage directly with our esteemed speaker. Please feel free to unmute your microphones and ask your questions, or if you prefer, you can submit them via the chat box. We welcome questions in both English and Sinhala languages.
There's one question about getting the HCPC registration. As I mentioned earlier, so I personally haven't done HCPC registration because I am a band 5 genetic technologist, and what I am aiming is to get a clinical scientist position, and because of that reason, I haven't personally done the registration in HCPC. But the thing is, it's really self-explanatory. I think the documents you need to so you would need the curriculum information from the university, and then it used to be like, I know a few people who got the registration saying that English is their first language, but things has happened, and now they really need you to show the IELTS qualification. I think the band 7 level, and then document wise, do you need the passport copy and address proof, degree certificates and service certificate. And then supporting information. I think you need the reference from where you are working to support what you have expressed in your application process. After you start your application in the website by creating the account and filling all the information, it's going to be really self-explanatory. It's going to guide you by step, and I think that's my knowledge about registration. But I will put my email address in the chat. If you guys have more questions, you can put them, and then I will try to answer them. I will try to do some research and answer them. Yeah, so now there are people who got their registration saying that English is their first language and they didn't do IELTS. But now I think so not even from people from Sri Lanka, like people from India, and a lot of people did that. But now for all the international students or applicant, they ask for IELTS. And you need 7, the score 7, and no score less than 6.5, I think. Oh, also, I would like to say you can write any questions in your in the chat box, or you can unmute and talk. The first two jobs, I found in UK, I found them through LinkedIn. So after you like you should start having a good LinkedIn profile. I found my first two jobs through LinkedIn, and if you connect with someone, just don't find them in Facebook, because Facebook is a private space. It's for friends, but it's very professional to have like a LinkedIn profile and keep it up to date and try to show your qualification and interest. So LinkedIn is a really good place. You can do some research on people and organizations, because if you are after a library manager or someone, you can see, and sometimes they post their job vacancies in LinkedIn. Also, they post them in the website I showed you before, but they also post them in the in LinkedIn post. And when you approach them, just don't ask for, oh, I have this qualification. Can you like give me a job or something? Don't ask that, because why would they give you a job? Show them some interest. Show them like, so if they have done a new test or something, or like if they have shared an article or something, read that article. Then probably you can tell them, oh, I am undergraduate, or I just graduate and I work in these things, and the thing you shared is really interesting and that inspired me. I have few questions. Would you like to have a little meeting with me, a Zoom meeting? 5 minutes would be appreciated. So try to build a connection, like a professional connection. Um, I know we all like instant result, but in real life, it's a bit competitive. Instant is not the way to go. Sometimes people are really lucky. Sometimes people get instant result, but I think just not believe in the instant pathway, but believe in yourself and try to build a process. So there's the one question about opportunity to work in UK without a CPC registration and directly apply to a job via those sites. Yes, that is possible because for some jobs, you like as I mentioned earlier, we have a tier system. So band 5 biomedical scientists need the registration. Band 7 clinical scientists need the registration. So in clinical team, up to band 7, you don't need the HCPC registration. But in band 6 level, you work toward the registration. That is the promise you give them when you get the band 6. I'm going to work on my registration. I'm going to get the registration. Until then, you work as a band 6. But to come to a band 6 clinical position, you need to have fulfilled certain requirement, like certain level of experience. So that's for band clinical team. For the technical team, so sometimes some hospitals, if they really need you, they might consider taking you. If you have right education qualification, if you don't have the registration, they might consider taking you as a band 5 biomedical scientist, and then they will give you the opportunity to gain the experience and then apply for the registration. That is possible, but because of the competitive nature of the jobs, that is kind of rare situation, but that's not totally impossible. Hmm. Oh, so there's a question about, is biomedical science and medical laboratory science degree are the same? I think the name of the degree does matter, but the most important thing is the curriculum. So when you get the registration, they would see your curriculum mostly, not the name of your degree. If you have covered all the areas needed for the registration, it doesn't matter name of the degree or the what university you go to. It doesn't matter if you have covered the right level of qualification, the curriculum. So there's a question about higher studies. What are the trustworthy agencies to apply? I think I never go through an agency. It is totally possible you to apply for higher education by yourself. You only need to invest 3-4 hours just reading their government website. The website is gov.uk. I will try to find the link. You can find any information you need, and of course you can go through an agency, but I really don't know any good agencies. gov.uk. So that's an interesting question. After finishing our degree, is it possible to get the job immediately if we migrate to UK, or how long it take to get a job? It totally depends. So I have few friends who got their job even before they finish their degree, and they transfer from student visa to work visa. And I know some people, they have finished their degree like 4 months ago and is still looking for a job. It totally depends on your portfolio. and based on like your experience and what kind of job you are looking for. But in majority, like I would say the NHS, the hospital job application process, they normally take 2 to 3 months. Okay. Is there any way to certify the knowledge on ISO through online platforms? I am not sure, but just talking about ISO is enough. Like in interview or like in uh, job application process, they want to know that you know about the laboratory ISO standard. Just I don't think they expect you to know all the details, but this is the standard we are working toward. I think that's the level they expect, but I personally don't know. But there's a few website, so let me find it. So there's a website called futurelearn.com. So most of the hospital, they collaborate. I put that in the chat, the link. They collaborate with different hospitals and then they have create different courses. I can't guarantee and I can't like promise you and I can't just promise you and tell like if you do this if you do any of these course, I can't just promise that you are going to get a job. But I have done like 4 5 because I didn't have any genetic experience. I did 4 courses in that platform, and because those courses are from hospitals, staff like clinical scientists, it impact in a good way to my application. Mm. Could you say something about GPA, madam? Okay, don't call me madam. Um, how to say? GPA sometimes it can be important, but I would so try really hard to get you a good GPA. It's a good thing, but it does not decide how you're going to improve in your career as a scientist. Because when I first joined university, my English knowledge was zero, and I failed like half of the exams in uni because I didn't understand a thing. And then it took me like very good time to get familiar with all the words and stuff, and it was really hard time. The first year was horrible because of my lack of understanding in language. But then that affects my GPA a lot. But now I experience, yes, the GPA matter. It's really good to have a good GPA. It helps. But that does not decide anything. You can work on it. If you have a low GPA in your bachelor, you can do a master, you can do a postgraduate degree and then top up things. But please try your fully to have a good GPA. So ASCP, it's not an exam. Is that the one I mentioned? AHCS. So AHCS is a registration process that's only for the genetic technologist. Which category we include after graduate as a medical laboratory scientist? So I don't fully understand the question, but I think like do it's like do you want to work in biochemistry, microbiology? It's totally up to you, because I know your degree covered really vast area. You study hematology, biochemistry, microbiology, everything. So find something inspired you, find something you really like to do, and then try to build on that, try to gain experience in those areas. So then you can step up in your career. Oh, reference books. No, like books are good, but there's a question about reference book that you recommend on molecular genetic and cytogenetics. Um, for the I would say I really don't know about books, but if you are interested in something in genetic, you can find research papers, and when you read research papers, see which year they are and if they are peer reviewed, and try to find good research papers. I think the books are amazing, but researchers, the new research articles, they give you like wider understanding. Because of most of the diagnostic lab, they refer back to researchers, and then if you refer back to researchers as well, the latest ones, you kind of have same level of understanding about things. So I think that would help than just reading a book. There's a question. Without considering GPA, can't we apply for those courses? Which courses? So any of the courses I applied, the GPA didn't matter. So there's a question, how to become a clinical microbiologist after finishing the degree. So after you finish your degree, so in this question, I think it's you want to be a scientist in microbiology, clinical microbiology. So that's what I understood. Please correct me if I'm wrong. So you need to have your experience in microbiology, and I'm sure your degree has basic solid knowledge on microbiology, and you can gain hand-on experience in clinical setting for microbiology, and then you can start probably working as a technologist and then upgrade within the. So that is personally my plan, but I'm not saying everyone has to do it, but there can be shortcut, there can be other ways. My experience is so, and it is going to be same for most of the disciplines. So I have my basic degree, and then I did a master in the field I think I want to pursue my career. So because of that, I did medical genetics. Then because of the then I start working as a band 3 in a genetic lab. So as I mentioned earlier, you don't need a master degree to work in a band 3 job, but just to get an experience, you can start at even lower band and then progress. And then if you want to go into clinical microbiology, then you have to broaden your understanding in clinical aspect. And then if you start, and then you would if you start working in a microbiology lab, then you would see, and you would get the opportunity to work toward the clinical team, and then you get the registration as a clinical scientist in specialized in microbiology. So when I mention you can get the registration as a clinical scientist, not all the registration are the same. So I can be clinical scientist in genetic. You can be clinical scientist in hematology, microbiology, biochemistry. So you can choose your discipline, and then you can become a clinical microbiologist. Yeah, of course. I think I put the email address. You can email me. That's a question about extracurricular activities. Um, when I say like extracurricular activities, what my mind go to is like doing volunteer work, things like that. Actually, doing volunteer work can be important. As part of my degree, we volunteer to work in a rural lab, and then we got the volunteer experience. So things like that help. Then it showed that you have like a giving personality, and they would see you as a good fit for the team. But I would say volunteer work is desirable. Quite, I mean, it's nice to have, but I would say most importantly, develop your communication skill, develop your skill in English language, and when you apply for a job, do your homework, be more confident. I think show up as the best person they could have in their team. And I think those are the qualities you should show them. The other things like volunteer work, this is a great topic you can talk in the interview or in your application process. But I think most importantly, your communication, your confidence is the most important things. Mm. So, is there any advantage of completing online courses such as bioinformatics? Um, y- like there is advantage, but do what is essential for you. So, because I like to continue my career in genetic. I like to do that is going to help me to accelerate my career, like try to find good courses. And but just doing that course just not going to give you huge advantage, but it is going to give you an advantage. Courses are great, but I think most importantly, in my experience, doing your own research is the best thing. There are a lot of research papers, research studies out there. I think that's the best tool. But of course, showing your qualification as an online course can help. Work experience on genetic. Is it okay to get experience as RA or TO? If I'm trying to go abroad to work as a... Okay. So let's say you want to work in different country in genetic, but you don't have that experience in genetic in your country in Sri Lanka. So what I would do is I can assure that you have all the solid knowledge you need to understand about things in genetics. And if you want to work as a TO or anything, that is fine. Everything is a good experience. Everything is a good opportunity to you to grow. Um, if you don't have any experience, that does not mean you can't apply or you can't get it. But what you can do is you can start at a lower band and then progress with your experience. I think that is what I would do. And also, LinkedIn is a great place. Try to connect with people through LinkedIn and show you are passionate, you are interested. Show them you are interested person because you are interested on what they are interested. Show them that. Studying in master in business administration or any relevant management master degree is helpful to laboratory management field. Okay. to get to that level. So laboratory managers are band 8. To get band 8, you need a lot of like years of experience. Most of the band 8 I know, they have like 25 years of experience in what they are doing. I'm not saying you have to have 25 years of experience, but that's the level of experience they have. So when they come to that level, the government give you the management degree. Like they give you, not the degree, they give you training in management, because you need to know how you're going to manage, what are the strategies you are going to use. So they give you training on management. If you already have a degree in business or administration, I'm not saying that's not an advantage, but if your aim is to go for a band 8 and be a lab manager, now you are spending time and your energy on a business degree is not gonna be that help if you are planning to go to this route. But if you want to go to administration job, like you want to be a project manager or something like that, that would help having a business degree. To get experience, is it okay to work in Sri Lanka for 2 years? Did you work directly in the UK in first job? Oh, yeah, of course. So all the experience matters. In here in UK, we have a lot of people with right qualification, but not with the right experience level they expect, and right understanding, the depth of understanding. So what you can do, it is totally okay to do work in Sri Lanka. I worked for 3 years after I graduate. Um, so that's going to help you, because then you build on your experience, and it's really helpful. I think I think that's the best thing I did, because I got a lot of experience. 3 years is not massive amount of years, but I got enough experience to answer the questions in my interviews, because most of the questions are more practical questions. Is embryology available in our field to become an embryologist? Um, I have no idea. But I had a friend when I did my master's. She was an embryologist when she was in India. She's from India, and then she did a medical genetic degree, and then she went back to she found a job in embryology in UK. How the genetic degree helped her is because uh, for the embryologist, they need to understand, they need to have a genetic basis as well. So I think that's how helpful it was to her. But I think it's possible. It's possible having a BSc in medical laboratory science is enough to be an embryologist. So a lot of people ask about extra courses that we can follow related to this field. You already have the highest qualification you could have. That is your bachelor degree. And if you are interested, if you're always motivated to learn, of course, do the courses. But personally, my advice is, because you already have the education qualification, try to gain more work-related experience, because then not everyone can get the work-related experience. So because of that reason, you're going to be highlighted in any application process. What is your best advice for fresh graduate? First graduate, I think try to get better experience in the field you are interested. Like try to find the I'm sure like most of the time when you learn through things, I have friends like really passionate about microbiology. They really like to draw on the plate and they are really passionate. They enjoy doing it. Try to find the field that you're going to enjoy doing and then try to decide like, do you want to go in academic side? Do you want to work in clinical side? Do you want to learn all the techniques and go through technical side? Like then you try to have a higher goal all the time. Try to have a higher goal, raise your standard. Because when I was in Sri Lanka, I didn't know my standard. I didn't know that I can do better. I didn't know my potential. After I exposed to a lot of jobs and after I see a lot of people, I realized, okay, I can do so much better than them. So you need to understand you have a lot of potential. Try to understand what you want to do and go full on. You can do it. Okay, where did you work? Are there any specific place to work in first job? Um, I don't think there's any specific jobs. So I think when I first graduated, I worked for a private lab and I was the phlebotomist, I was the technologist. I had to write the bills. I was just one person. It was one man station and it was a private, not a hospital, just just a pharmacy or something. And then that was my first job. And then I did work there for like 3 months until I get my first permanent job. But the thing is, I learned how to troubleshoot all the machines I had to work with because I was the only person to do. So when I worked there, I used to look down on that because everyone working in nice, good jobs, and I still couldn't find a job. Then I was like, but I learned how to draw blood from patients, and I draw blood from like 500 people within that 3 months, and now I am really proud of the experience. Um, I had that time, so any job is gonna build you on what toward your goal. Any job is going to help you. Then I work for the Jayewardhanapura hospital for a few years, and then after I start my master's here, and then when I start during my studies, I work for a private hospital in London, and because that was the COVID time, they couldn't have a lot of people. Sometimes there's no one to draw blood, and then I can step up and say, okay, I have experience in phlebotomy, and I can do that, and then I became like an essential part of their team. So when I do the phlebotomy, when I was doing one man station, I was looking down on myself, but I gained the experience. And all the experience you get, it's gonna help you in some way someday. Okay, bioinformatics after MLS degree. So bioinformatics is really, really, really interesting field. And we have a bioinformatics team in our hospital as well. It's a great field. And I think I don't have a lot of understanding about bioinformatics, but if you can send me email, I will try to find information. I think you need to have some level of educational qualification. I know in my master's program, the program I did was medical genetics, and we have a 3 month of bioinformatics course during that program. And one of the students, she works in Canada as a bioinformatician after this program. So you need some level of education qualification, and then you have to get the work experience because it's a new field and it's a really interesting field. What do you recommend for work experience for genetic field, private sector or government sector? It doesn't matter whether it's private or government. What matters is what you gain from it and how you present yourself. That is the most important things. So like learning karyotyping in a private hospital or a government hospital, if you represent that, if you say that, oh, I did karyotyping in this hospital, they are not going to know the name of the hospital, but they are going to recognize your experience. And based on your experience, they would ask you questions. So if you say, I did karyotyping in private hospital, and then they would ask, oh, what kind of karyotyping? If this happened, how would you troubleshoot? So if you can answer that question, it doesn't matter where you worked. That's same for the phlebotomist courses. If you can do your job, it doesn't matter, private or government, it doesn't matter. Is it necessary to study IT degree to engage with laboratory technical side? No, you don't need IT degree. In my experience, people who work as a laboratory technical side. So when you say laboratory technical side, I hope you mention the laboratory staff. We don't need an IT degree, but you need some level of understanding. If you have another registration, good to mention when apply. Of course, you can mention that, but I wouldn't say they will give marks based on that. Like if you have a registration in another country, like we all have registration in Sri Lanka, they never give like any priority based on that registration. But if you already you don't have to apply for that. If you already have it, you can mention that because it could give you some kind of unofficial recognition, but nothing official. Hmm? To become a researcher in bioinformatics, does the experience on laboratory work matter? It doesn't harm, but I think if you want to go to bioinformatics, it's okay to not to have experience in laboratory work. Because it's in bioinformatics, you do desk-based job. Most of my bioinformatics team does is we have sequencing, NGS sequencing results. Most of the time, what they do is they They make the results more presentable. Actually, to be honest, I have limited understanding about bioinformatics. Only thing I know is it's really interesting because you know everything about how they work, all the data and data manipulation, and with the AI and everything, bioinformatics is going to be a blooming field in next probably 5-10 years. What is the least position we can apply only with BSc degree? So if you want to work in technical team, the least is band 5. Um, and if you are going in clinical team, band 6. When you go to band 6, you need a master degree. But if you don't have a master degree, up to band 5 level, you can apply. Of course, your undergraduate research will help you in your future career because it will show you can do a research. You understand research methodology. You understand how to do the literature search. You understand how to do the references. You read everything. You know what to do. So it's definitely going to help. So put good weight on that. And one thing I want to tell you guys is I think we all should raise our standard. Just don't look for the least, look for the highest because you all have the potential to get that. I think just look for the highest where you can go and work toward that. Have a long-term goal and work toward that. And also, there can be some people. So I had friends who really want to go abroad and work. And I also still have friends who really don't want to go outside the country and they want to live with their family, and because of some personal reasons, they have to stay in the country. But um, see your potential and try to so one of the reason this can be a little bit of controversy. So some of the graduate, they look for the recognition, a better recognition in their career. So it is our responsibility to build our recognition in our career. We can do more. We should raise our standard. When there are no more qualification, us to continue when there are no more questions in. Okay, there's a one question about master, MPhil, and PhD. So where I found my masters, I will try to find the link and put in the chat. Mm. I don't know why I can't send you this link. Okay, so I have put a link. So that is where I found masters. But the easiest way you can do is like if you want to study in London, look for universities in London, and it's really simple. You really have to like spend a few minutes browsing through their website, the university website. This is how I did. I did found a lot of like masters through this link I provided. Because it's like the collection of all the universities and degrees, and then I listed a few universities or courses that I can afford because that's important, and I am interested. And then what I did is I had like 3 4 universities that I like to go. I like to study in, and then I email the so when you find a university, then go to the course in university website, and then try to like build a contact with the course provider, because they normally put their email addresses and everything about the course provider. Then you can ask them like for like advice. It's really good to build on your own work. Okay, can you please tell something about undergraduate research? I don't have any idea about that. So I think probably it's different from university to university, how the undergraduate degree would go. Um, Tell something about undergraduate research. So I think so your university will tell you, or sometimes I think you can choose, but based on the resources you have, I think they will tell you, or like probably if you're lucky, you would get a chance to choose what you're going to do on your research. And I think thoroughly put a lot of effort in learning how to do a research. I know we all want to have the best results and come up with best conclusions, or the world best discussion and stuff, but at undergraduate level, my understanding is you really need to know and learn and get experience on how you are going to do a research. What is the research methodology? Why is it important? How we gonna, if I have to do another research one day, if I want to be a researcher, how this experience is going to help me to get me to that level. So I think it's better to focus. It's really great to have the best results, but best discussions and conclusions and world renewing information. But at the same time, it is really important you to uh, learn about all the methodology and technology. Like it's good to have the best discussion, but it's really good to have really good to learn how to write a discussion. So things like that make it like a good learning process. Of course, do your research best to your capabilities. Try to get the best results. It is like sometimes the result for my research, I got the results does not match what we wanted to tell. So that's okay. Then you can tell like, oh, these things happen. Probably this is why we couldn't get these results. This is the expected result. So it's going to be interesting. You will enjoy. No problem. It was really amazing experience to talk to you guys today. I feel like I did something good for what I got. And it's really nice to see a lot of engaging messages.
So this is the end of our Q&A session. Thank you all for the thoughtful questions and the lively discussion. Before we proceed further, I kindly request you all to turn on your videos so we can take a group screenshot to commemorate this moment. Thank you all. As we approach the end of this session, I would like to invite Mr. Ravindu Jitmal, the Vice President of Ruhuna Medical Laboratory Science Student Society, to deliver the vote of thanks. Mr. Ravindu, over to you.
Good evening all. Today our guest speaker Ms. Dananjani Rathnayake and all the lecturers and my dear colleagues. First of all, I would like to thank for giving this opportunity to deliver the vote of thanks of the second webinar of this year. Also, my special thank is given to today our research person, Mrs. Dananjani Rathnayake, for conducting today's webinar on unlocking career opportunities in UK by devoting her valuable time on behalf of our special request in order to explore career opportunities for MLS graduates in the UK and discover exciting career pathways. So thank you very much once again on behalf of all the participants and MLS Society. And also I'd like to thank Dean of Faculty of Allied Health Science, Professor Imendra Kotapole, Head of Department, Dr. J.A.N. Sadamali, Dr. Sachinthi Amarasiri, and Dr. Eranga Silva for empowering and encouraging us to organize this webinar. And also all the members of MLS Society who worked hard to organize this event. And last not least, all the participants who are always motivate us to organize more and more events like this. Also, I would like to express my apology if there are any mistakes while this event, behalf of MLS Society. So thank you very much. Have a nice day.
Thank you, Mr. Ravindu Jitmal, for those kind and appreciative words. With that, we officially conclude the second session of our webinar series. We hope that today's discussion has offered you valuable insight and sparked your curiosity about career opportunities in the UK.
Thank you all for joining us today, and we look forward to welcoming you to the next session of our webinar series. Until then, take care and have a wonderful evening ahead. Goodbye.